Deep in the heart of Alabama, a silent threat has emerged from the shadows. A virus, previously unknown to science, has been discovered lurking within the tiny bodies of American shrews. This newfound pathogen, named Henipavirus, has sent ripples of concern through the scientific community. It serves as a stark reminder of the invisible world of viruses and their potential to impact human health. The discovery, like so many in the world of emerging diseases, was a twist of fate. Scientists weren't actively searching for a new virus, but sometimes the most important discoveries happen when we least expect them. Routine surveillance in a small population of shrews unearthed something unexpected: the telltale genetic signature of a completely new virus. The finding was both exhilarating and concerning. While the discovery of any new life form is a scientific triumph, viruses with their potential to cause disease always spark concern. The next step was clear: to learn everything they could about this enigmatic virus and the threat it might pose. The clock was ticking. Henipavirus belongs to a family of viruses known as henipaviruses. This family already boasts some notorious members, including Henipavirus and Henipavirus. These are not names whispered lightly in scientific circles. They are harbingers of severe disease, known to cause encephalitis, a dangerous inflammation of the brain, and respiratory illness in humans. Henipavirus, first identified in Malaysia in 1998, erupted from pigs to people, leaving a trail of devastating illness and fear in its wake. Henipavirus, discovered in Australia in 1994, jumped from bats to horses and then tragically to humans. Both viruses are zoonotic, meaning they can spread from animals to humans, making their emergence a serious public health concern. The Henipavirus family is known for its broad host range, its ability to infect a variety of animal species, and its high mortality rate in humans. Some Henipaviruses are even considered potential bioweapons due to their virulence and the lack of effective treatments. The emergence of a new Henipavirus, Henipavirus, raises immediate questions about its potential to follow in the footsteps of its deadly relatives. Henipavirus is a relative newcomer to the scientific stage, and much about it remains shrouded in mystery. Scientists are working tirelessly to piece together its secrets, to understand how it spreads, the symptoms it causes, and its potential to infect humans. Early research suggests that Henipavirus shares some similarities with its Henipavirus cousins. It possesses the same genetic machinery, the blueprint for replication and spread, that makes Henipaviruses so adept at jumping between species. However, every virus is unique, and Henipavirus is no exception. The behavior of a virus, its ability to cause disease, and its preferred hosts is dictated by a complex interplay of factors, many of which remain unknown for Henipavirus. Scientists are particularly interested in whether Henipavirus shares the same predilection for neurological and respiratory systems as other Henipaviruses. The answer to this question will be crucial in understanding the potential threat posed by this new viral enemy. The discovery of Henipavirus in Alabama shrews has thrown these tiny, often overlooked creatures into the scientific spotlight. Shrews, small mammals with long snouts and tiny eyes, are found throughout the world, playing an important role in their ecosystems. Dr. Rhys Parry, a leading virologist at the forefront of Henipavirus research, and his team at the University of Queensland are at the forefront of this investigation. Their work centers on understanding the relationship between the virus and its shrew hosts. Are shrews merely an incidental host, or do they play a more significant role in the virus's life cycle? The team's research has revealed that Henipavirus is widespread in shrew populations in the southeastern United States. This finding raises concerns about the potential for spillover, the transmission of the virus from shrews to other animals, including humans. The race is now on to determine how easily the virus can jump species and whether it poses a direct threat to human health. Section 5. A continent on alert. The implications for North America. The discovery of Henipavirus in North America has put the continent on high alert. While Henipaviruses have wreaked havoc in other parts of the world, this is the first time a member of this deadly family has been found on North American soil. This finding has forced a reassessment of the risks posed by emerging viruses in North America. For too long, the threat of Henipaviruses has felt like a distant problem, confined to the tropics of Southeast Asia and Australia. Henipavirus has shattered this illusion. The presence of a Henipavirus in North America raises the specter of a potential outbreak, a scenario that sends chills down the spines of public health officials. The United States, like many countries, is ill-prepared for a widespread outbreak of a deadly and highly contagious virus. The discovery of Henipavirus serves as a wake-up call, a stark reminder of the importance of investing in public health infrastructure and disease surveillance. Section 6. Spillover threat. Could Camp Hill jump to humans? The million-dollar question that haunts researchers studying Henipavirus is whether it has the potential to jump to humans. While the virus has not yet been found in humans, the possibility of spillover cannot be ignored. History is littered with examples of viruses that have made the jump from animals to humans, often with devastating consequences. The HIV/AIDS pandemic, the SARS outbreak, and the COVID-19 pandemic are all stark reminders of the ability of viruses to cross species barriers and wreak havoc on human populations. The close association between humans and wildlife, coupled with the ever-increasing encroachment of human activities into natural habitats, creates fertile ground for the emergence of zoonotic diseases. The more we learn about Henipavirus, the more we realize that we are in a race against time to understand and mitigate the risks it poses. Section 7. Inside the lab, decoding the viral enemy. In laboratories across the globe, scientists are engaged in a high-stakes battle against an invisible enemy. Armed with the tools of molecular biology, they are dissecting Henipavirus piece by piece, unraveling its secrets in a bid to stay one step ahead. Virus identification and analysis are complex processes that require sophisticated equipment and expertise. Scientists use techniques like polymerase chain reaction, or PCR, to amplify viral genetic material, allowing them to detect even minute quantities of the virus in samples. Once the virus is isolated, scientists can sequence its genome, revealing its genetic blueprint. This information is crucial for understanding the virus's evolutionary history, its relationship to other viruses, and its potential to cause disease. Scientists are also working to develop diagnostic tests that can rapidly and accurately detect Henipavirus infection, a crucial step in controlling the spread of the virus. Section 8. The race against time, vaccines and beyond. The discovery of Henipavirus has ignited a race against time, a global effort to develop effective vaccines and treatments before the virus can claim its first human victim. Vaccines, by mimicking natural infection, prime the immune system to fight off a specific pathogen, providing a powerful tool to prevent disease. Developing a vaccine is a complex and time-consuming process. Scientists must first identify the parts of the virus that trigger an immune response known as antigens. These antigens are then used to create a vaccine that tricks the body into thinking it is infected, triggering the production of protective antibodies. While researchers are working tirelessly to develop a Henipavirus vaccine, there are currently no approved vaccines or treatments available. This underscores the importance of continued research and investment in vaccine development, not just for Henipavirus, but for other emerging infectious diseases as well. Section 9. Global Health at a Crossroads. The emergence of Henipavirus serves as a stark reminder that we are living in an era of unprecedented global interconnectedness. The movement of people, animals, and goods across borders has created a complex web of interactions that can facilitate the rapid spread of infectious diseases. In this interconnected world, the health of one nation is inextricably linked to the health of all nations. The discovery of a new virus in one corner of the globe is a concern for everyone. The fight against emerging infectious diseases requires a coordinated global response. This includes investing in disease surveillance, strengthening public health infrastructure, and promoting international collaboration in research and development. The emergence of Henipavirus is a wake-up call, a reminder that we must remain vigilant and